talk about Chapter 8, which is basically about cellular reproduction or cellular division or cellular replication. They're all the same thing. And then this actually involves two different types of process. It involves mitosis and meiosis. I know, right? They sound pretty much the same thing. But you guys will have to know how to compare and to how to contrast these two together. And then that's one of the things that may be difficult for students is to keep, what's happening in mitosis and what's happening in meiosis in line. So what I suggest you guys do is make charts to keep your information organized, or draw pictures, or look on other videos. Also, another difficult thing about this chapter, besides it's going to be a little bit long, but is you guys will have to know the number of chromosomes versus the number of chromatids in each phase of mitosis and meiosis. And then we're going to talk about exactly what chromatids are and exactly what chromosomes are. I know we've touched on it a little bit in the past. So there's going to be some examples of different species having their number of chromosomes, and then you'll have to know, for instance, what is the number of chromosomes or chromatids in prophase for a species that has a total number of chromosomes of 8. Okay, the example in your lecture notes is a good example. Please do it. I highly suggest that. Okay, so let's start with what cell division is. We know that cell division is when cells replicate so that we can either make a new organism or a new part, or just another cell. There's two types of cell divisions. We have asexual reproduction. Okay, so asexual reproduction is when you do not need a mate or do not need a partner, and then the offspring or the new cell or the new part is genetically identical to the parent. And then we have some examples. So here, this is actually a picture of a yeast. Here we have the parent cell. So you see that the parent cell, it doesn't need a mate, so it's undergoing asexual reproduction by budding off a daughter cell, the new offspring. So for this asexual reproduction, you're actually making a new organism. And you guys will see that bacteria, we are going to make a new organism, a new bacterium. Here, another good example for asexual reproduction is that a starfish, it can regenerate itself as long as it has the central disc in the middle right here. So if one of its arms were to sever off, and it can actually sever it off itself as a defense mechanism from a predator. But it's okay, because when it undergoes asexual reproduction, it can regenerate this arm, part of this organism, and then become whole again. And then another good example of asexual reproduction is our liver cells or our liver organ. And many of our cells actually can undergo asexual reproduction. But the liver is the only organ in our body that it can be missing just about 50% of its part or whole part, and it can still regenerate and function as normal. That's why you hear, people can donate a lobe of their liver, and then that donor would still be okay because within a few weeks, their liver will regenerate under asexual reproduction and become whole again. Now, if the donor is drinking, of course, they wouldn't be a donor if they're drinking, right? But if somebody was drinking, they would not be able to donate because their liver wouldn't have that capacity. They don't have those healthy cells to have that capacity to regenerate as well as a healthy person. So what are some advantages and disadvantages of asexual reproduction? I'll give you guys a couple minutes to think about it, and then we can talk about it. So let's start with advantages for asexual reproduction. if the environment is stable, then that's good because the parent cell will make offspring that are genetically identical, the same DNA, the same characteristics, the same traits, and then those offspring will survive in that stable environment because they have the genes to um, survive, reproduce. And then another advantage is that asexual reproduction, you don't need a mate, you don't need a partner, okay? Another advantage is that it takes less time, so you can reproduce faster to get a large number of offspring. And then also another advantage is that it takes less energy, so less resources of the cells to replicate itself or the organism, which is always good. Well, the disadvantage is that, if you are able to produce faster and then produce a large number of offspring faster, then that means that there's more competition for the resources in that stable environment. Okay, and then what happens if that stable environment no longer is stable and there's a change, there's like a climate change or something's happening, a disaster? Well, then they don't have the genes to adapt to that change in the habitat in their environment because they don't have the genetic diversity like we do. So if they don't have any genetic diversity, their gene pool is always the same. They'll always have the same traits. They will not be able to have the genes to adapt to a new environment. And then if there's no genetic diversity or genetic variation, then evolution may not occur for these types of organisms. But of course, if we have a mutation, then we would have some kind of evolution, but most likely not. And then the other type of cell division is sexual reproduction. So sexual reproduction pertains to humans, us, and animals, of course. And then we can see in our society that, when we undergo sexual reproduction, our offspring, our children, are not genetically identical. So if you're a parent and then you make some children, your children are not going to be the same as you. They're not going to have the same DNA, the same exact DNA. There's going to be some genetic diversity. It's a combination of the parents to make the children. So what are some advantages and disadvantages? So a disadvantage would be that we would need to find a mate, a partner, to undergo that sexual reproduction, to make offspring. And then sometimes that can take a long time. And then, of course, we have a long gestation period. For humans, it's 9 months. I think in elephants, it's 2 years. So that means that we don't have the capacity to make as many offspring as an organism that undergoes asexual reproduction. Bacteria, they can replicate very, very fast, and then that's how you can get food poisoning in just about 4 to 6 hours. And then an advantage of sexual reproduction is that we do have genetic diversity. We see it in us, we see it in dogs. Okay, our gene pool is very diverse. We have many traits, we have many characteristics. You can see it, everybody's facial appearances, everybody's personality traits is very different. So this genetic diversity, if we were to have a change in our environment, we have the genes to adapt. We will most likely not become extinct for that reason, and then our possibility for evolution is more favorable than asexual reproduction. And then here we see that, there's a sperm, which comes from the father, an egg, which comes from the mother, gets fertilized by that sperm, and then what forms is a zygote. and then the zygote will eventually replicate, and then it'll become a fetus, and then a baby is born. So here, this is a picture of my doggies. This is Abby, this is Buddy, and then they were formed from asexual reproduction from their parents. So they are different from their parents. They are not genetically identical to their parents. Let's talk about how bacteria or prokaryotes replicate. They undergo asexual reproduction. They undergo what's called binary fission, and then just means that it's splitting in two. So what happens is that here we have a bacterium, and then for reproduction to occur, the DNA, which is just a single circular chromosome, it has to replicate or duplicate. Now, why is that important? Well, if it splits into two, that means each bacterium will need its own set of chromosome or its own chromosome to survive. So we have the chromosome or the DNA replication, and then we have their plasma membrane, cell wall. It'll elongate and eventually separate the two bacteria. And then now we have two daughter cells that came from this one parent cell. So these two daughter cells are genetically identical to the parent cell, which means that it just has the same DNA sequence and it'll be exactly the same as their parent cell. Okay, so some review before we go into eukaryotes. So you guys remember what chromatin is versus chromosome? Now this chapter will have a lot of C words. So that's another thing that may confuse students. So maybe it's good to make flashcards to know what the definition of these new terminology words are. So chromatin, okay, here, this is what chromatin would look like. It's just this loose strand of DNA or loose molecule of DNA. And then chromatin is the state of DNA for the majority of the cell when the cell is not dividing, when it's carrying out its normal functions. And then on the DNA, on the chromatin, you have these histone proteins attached to it. And then what these histone proteins do is that they will coil the DNA around it to condense the DNA, pack it into the state of chromosome. And then when the DNA is in the form or state of chromosome, that's when the cell is about to divide. And then these pictures here, these are just some pictures of something that's called karyotypes. And then karyotypes are just a visual display of the chromosomes in a cell. So here you have a diploid, which is 2N, and then in us, we have 46 total chromosomes in our body cells. So we have a pair at chromosome 1, a pair at chromosome 2, a pair at chromosome 3. This chromosome may have come from mom. This chromosome came from dad. This chromosome came from mom. This chromosome came from dad, etc., etc. And then here you have your sex chromosomes. So this X chromosome came from mom, and then this Y chromosome came from dad. So this human karyotype or this cell is from a male because of that Y chromosome. And then how do we get this diploid cell? Well, it comes from a haploid cell and another haploid cell. So here, this is a karyotype of an egg cell. And then you notice that the egg cell, it only has 23 chromosomes. It does not have pairs. It does not have a pair of chromosomes at chromosome 1. It only has one chromosome at chromosome 2, one chromosome at chromosome 3, etc. And here we have the sex chromosome of the egg. Females can only donate an X chromosome because females are XX. Whereas males, they are XY. So they can donate an X chromosome. In this case, the sperm is going to donate a Y chromosome. So here, the sperm cell, it only has 23 chromosomes as well. It doesn't have any pairs. So it has one chromosome at chromosome 1, one chromosome at chromosome 2. Okay, so this is from the mom, this is from the dad. And when the sperm fertilizes the egg, it becomes a diploid cell. It becomes a diploid cell. So this chromosome came from the sperm. This chromosome came from the egg, sperm, egg, and now you have 23 pairs of chromosomes for a total of 46 chromosomes. 46 chromosomes. And don't worry, we're going to repeat all of this. Okay, so this is what I would normally write on the board. So some definitions. Diploid cells are what's called your somatic cells. Somatic cells. And then these somatic cells are your body cells, for example, your skin cells, your eye cells, your liver cells, your stomach cells. And then these somatic cells or diploid cells, the way that they're created is through asexual reproduction, through mitosis. Mitosis. And then diploid cells are always denoted 2N. 2N. And then the 2 represents the number of sets of chromosomes. So there's 2 sets of chromosomes in a diploid cell. And then the N represents the number of chromosomes in that set. So for us humans, we have 23 chromosomes in each set, but we have 2 sets of chromosomes. So what do we do with those numbers? We multiply them. And then we get 46 chromosomes total in a diploid cell. So that's how we get that number 46 chromosomes. There's 23 chromosomes in each set. So that means there's 23 chromosomes in an egg, there's 23 chromosomes in a sperm, and then that represents 2 sets. We multiply that together to get 46 chromosomes. 46 chromosomes. And then mitosis, a summary of it is that we start off as diploid, and then we undergo DNA replication because each cell will need its own sets of chromosomes. And then after DNA replication, we still have that diploid number. So we'll have 46 chromosomes, 46 chromosomes. And then after mitosis, after replication, we end up with 2 daughter cells that will have 46 chromosomes each, that are diploid each. And then these daughter cells will be genetically identical to the parent cell. Because it's asexual reproduction. We want to make the same exact cells. So for example, skin cells will undergo mitosis. Our skin cells are diploid cells, and when they undergo mitosis, when they undergo reproduction, we want them to be exactly the same as the parent cell. We want all our skin cells to be exactly the same. Here we have what's called haploid cells. And then these haploid cells are your gamete cells, which are your sex cells. And then these sex cells are your egg cells in females, and then your sperm cells in males. And then these are produced by what's called meiosis. And then meiosis is for sexual reproduction. These haploid cells are denoted by 1N or just N. So that means that we only have one set of chromosome, and then for us, each set has 23 chromosomes in it. So what do we do with these numbers? We multiply the 1 times 23, which just equals to 23 chromosomes. So a sperm that has 23 chromosomes fertilizes an egg that has 23 chromosomes to make a zygote that has 46 chromosomes. We restore that diploid number, that 2N number, after fertilization. And then here, this is a summary of meiosis. So in meiosis, it'll start off with the diploid number, 2N. So it's 46 chromosomes for us, the human species. And then the DNA replicates, but we still have 46 chromosomes, 2N number. And then meiosis is a little different, it's special. There's 2 cellular divisions. We have a meiosis 1 and a meiosis 2. So here, after meiosis 1, we end up with haploid numbers, 2 daughter cells that are haploid numbers. That means that each of these daughter cells will have 23 chromosomes each. But it's not in the correct form, or there's still too much DNA. And then I will explain that in more detail later on. So that means that we have to undergo another cellular division, which is meiosis 2. Each of these daughter cells will undergo meiosis 2. So this daughter cell will produce 2 daughter cells that will still be haploid cells, but the DNA is the correct amount. This daughter cell will undergo meiosis 2 and still be haploids with 23 chromosomes each. And then the amount of DNA is correct. So in total, after undergoing meiosis 1 and 2, you get 4 daughter cells that are haploid each with 23 chromosomes, and then it will produce 4 genetically different cells, or 4 genetically unique cells. Whereas in mitosis, you produce 2 genetically identical cells. So this is asexual reproduction, this is sexual reproduction. Okay, so some more background information. So here, this is kind of what represents a chromosome. And then this is the chromosome before the DNA replicates. So for us, we have 46 chromosomes, which is diploid or 2N. And then we go through a phase called synthesis phase or S phase. And then this is just the DNA replicating. So that means that this chromosome will be replicated, and then here is the copy of it. So this X represents a duplicated chromosome. And then each of these duplicated chromosomes have what's called sister chromatids. So this blue is a sister chromatid that is identical to this sister chromatid. So that's like a reddish brown color. So this one duplicated chromosome has two sister chromatids. They're held together by a band of DNA called centromere. Centromere. So this little dot represents the centromere that holds these two sister chromatids together. So this whole thing right here, that's one chromosome. This is also one chromosome, but this is in the unduplicated state. This is the duplicated state. And then after S phase, you still end up with 46 chromosomes, but you have 92 chromatids because each chromosome has two chromatids. So basically, you're taking that 46 number, multiplying by 2 to get the 92 chromatids. Over here, you do not have any chromatids. You just have chromosomes. So since it's 46 chromosomes, you are still in that diploid state, 2N state. And then you undergo mitosis. So the point of mitosis is basically to separate these sister chromatids. So after mitosis, these sister chromatids, after they separated, they become individual chromosomes. So this is a chromosome, this is a chromosome, but they're genetically identical to each other. So what basically happens is that this chromosome is going to go to one daughter cell, this chromosome is going to go to another daughter cell. That's why you get two genetically identical daughter cells. So for us, our numbers, each daughter cell, after undergoing mitosis, will have 46 chromosomes, which is the diploid state. The other daughter cell will have 46 chromosomes, which is also, again, that diploid state. So this daughter cell will be genetically identical to this daughter cell. So let me ask you a question. If dogs have a total of 78 chromosomes, what is its haploid and diploid number? And then I'll give you guys 2 minutes. And then here we have a karyotype of a dog. They have more chromosomes than us. So for dogs, the diploid number, that 2N number, is 78 because they have a total of 78 chromosomes. And then to find the haploid number, it's just half of that, which is 39. So its haploid number is 39. So 39 chromosomes from the egg comes together with 39 chromosomes of the sperm. They fall in love, they mate, and then you get the diploid number of 78. Okay, so next we're going to talk about the cell cycle. The cell cycle is just a sequence of events that the cell goes through when it's about to divide and when it's dividing. And then it's generally categorized into two major phases. We have what's called interphase, and then we have the mitotic phase, which will include mitosis and cytokinesis. let's talk about interphase. So interphase is basically a long period of time when the cell is preparing to divide. And then in this interphase, basically the cell is in what's called a growing stage, is when you have to duplicate all the organelles, proteins. It's important to duplicate the organelles because you want both of the daughter cells to have its own set of mitochondria, its own sets of Golgi apparatus, etc., etc. We don't want, any sharing. We want, each cell to have its own set of organelles. So let's go through these phases. Here we have what's called G1 or first gap. And then this is when you are duplicating or replicating organelles and making proteins necessary for mitosis. And then here, S phase or DNA synthesis phase, this is when the DNA is just replicating. So you'll have two sets. You'll have a complete set for one daughter cell and then another complete set for another daughter cell. And then here, G2 or second gap, this is when you are replicating the centrosomes. And then remember, centrosomes are made up of two centrioles. So each centrosome has two centrioles. So this is one centrosome. This is another centrosome. So this centrosome right here, it has two centrioles. This centrosome has two centrioles. So normally our cells, when it's carrying out normal functions, it only has one centrosome with two centrioles. But when it's dividing, that centrosome will replicate. So we end up with two centrosomes, each having two centrioles. So that means we have a total of 4 centrioles when we are dividing. And then now we're going to make some more proteins, and then we are ready for mitosis. So, and then mitosis is actually when the cell is dividing. And then cytokinesis is when the cytoplasm is dividing. And then here, this G0, or sometimes called G0, that is just when the cell is carrying out its normal functions, when it's not about to divide or getting ready to divide or dividing. And then this slide is just to have a summary of the mitotic phase, which includes mitosis and cytokinesis. So mitosis, we've talked about here, we start off with one parent cell right here. And then we're going to end up with two separate daughter cells that are genetically identical to this parent cell. And then mitosis is basically the division of the nucleus. And then these are the phases for mitosis. So I do want to mention that some books will separate prophase and prometaphase. Okay, so some books will have prometaphase, but some books will only have prophase. So for our exam and for this class, we will not have a prometaphase. We'll just group this all together as prophase. So all of this will just be prophase. And don't worry about early prophase, prophase, or late prophase. On the exam, we would just ask you, if it's prophase. And then the next phase would be metaphase, anaphase, and then telophase. So these are the 4 phases for mitosis. Cytokinesis is when the cell is actually going to separate to become individualized. So it's the division of the cytoplasm. And then I do want to mention that cytokinesis is not part of mitosis. It is separate from mitosis. Okay, before we go into the phases, let's talk about the three functions or the three purposes of mitosis. Why do we even have mitosis? So one reason is that we have mitosis for growth and development. So all of us, we start off with one single cell, the zygote, and then the zygote undergoes mitosis. It replicates to become a ball of cells, and then we become an embryo, and then a fetus, and then we become a full-fledged human person. So we need mitosis so that our cells can replicate, so that we can have tissue. So remember, tissues are a group of cells. And then another purpose is to replace old or damaged cells. And then a really good example is your skin cells. So, we're pretty abrasive with our skin cells, but our skin cells have a very high capacity for regeneration, for mitosis. So that means that, it's okay that our skin sheds every 2 weeks or 14 days because our skin cells are constantly undergoing mitosis to replace those old cells. My husband just had a corneal scratch. Okay, so that's the front part of your eye, but your corneal cells, they have a pretty high capacity for mitosis regeneration. So that means that his cornea is okay because of mitosis. Those missing cells that he scraped, the injury, is able to replace those missing cells or damaged cells. And then another purpose is for asexual reproduction, to make genetically identical cells. So it's important to differentiate between asexual reproduction and sexual reproduction for mitosis. So remember, mitosis is only for asexual reproduction. So which types of cells do not undergo mitosis? Of course, your sex cells, your gametes, your haploid cells. But what other types? We have red blood cells. So our red blood cells, once it enters the bloodstream, it loses its nucleus. So if it doesn't have a nucleus, it doesn't have any DNA, and then how can it undergo mitosis? It doesn't need to. Its one function is to pick up oxygen, or to drop off oxygen and pick up carbon dioxide to our tissues. Other types of cells do not undergo mitosis. We also have brain cells or nerve cells. Nerve cells don't have the capacity to undergo mitosis, very, very limited capacity. So that's why spinal cord injuries or brain damage is so detrimental. People do not recover from those very well. You have paralysis, motor speech problems, etc. because these brain cells, these nerve cells cannot replicate. And then also, if you have a heart attack, so when you have a heart attack, those heart cells die, and then they don't, the ones that are still alive, they do not undergo mitosis. So what happens is that those dead cells are replaced by collagen fibers. And then if you have collagen fibers, the heart can't beat as well or contract or pump blood as well as regular heart cells, cardiac cells. That's why people who have heart attacks, it takes a very, very, very long time to recover, even, if they ever recover from a heart attack. So those are just some examples of cells that do not undergo mitosis. Let's talk about some parts before we talk about the actual phases of mitosis. So here we talked about it before. So this right here is a centrosome that is made up of two centrioles. So you have one centrosome here, one centrosome here. And then coming out, radiating or originating from these centrosomes, these centrioles, you have microtubules. So remember that cytoskeleton? These are microtubules, or we can say mitotic spindles, or we can actually say spindle fibers. It all means the same. And then these spindle fibers, what they do is that they connect to a protein on the chromosome called kinetochore. Kinetochore. So when these microtubules attach to these kinetochores, that's how they guide the chromosomes where they need to be during each phase of mitosis. And then of course we talked about this right here is one sister chromatid, and then right here, this is another chromatid. And then these two chromatids are connected, held together by a centromere, a centromere. Okay, so now let's talk about mitosis. So the first phase is called prophase. This will be the longest phase because there's so many things going on. So here we have our centrosomes, two of them with a pair of centrioles each. What's going to happen is that they are going to migrate or move to the opposite sides of the cell or opposite poles of the cell. And then you get the formation of your spindle fibers. You have your nucleolus, which will start to disappear. And then you have your nuclear membrane, which will disintegrate or dissolve, disappear. And then also you have your chromatin form of DNA. It will start to condense into chromosomes, So in prophase, you have 46 chromosomes. So each of this is a chromosome. You have 46 chromosomes. And then each chromosome will have two chromatids. So you have a total of 92 chromatids in prophase. The next phase is called metaphase. And then this is just when the microtubules are guiding the chromosomes to the center, the middle, or the equator of the cell. And then when it does that, that's called the metaphase plate. So these chromosomes are aligned on an imaginary line called the metaphase plate. But you didn't do anything to the number of chromosomes or the number of chromatids. You're just moving them around. So in metaphase, for us humans, you have 46 chromosomes and 92 chromatids. So the same as prophase. And then this metaphase is actually a checkpoint. The cell will ask the question, oh, are the spindle fibers attached to the kinetochore, the chromosomes tightly, securely? Are the chromosomes aligned correctly on the metaphase plate? And then if they're not, then the cell will give itself some time to correct those. The next phase, you have anaphase. And then anaphase is when you are just separating the chromatids. Separating the chromatids. So remember, these chromatids were together in metaphase, but in anaphase, you've separated them, and now these chromatids, they have become individual chromosomes. So now this is a chromosome, this is a chromosome, 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 etc., etc. In order for your chromatids to split, the centromere that was holding them together has to split as well or come apart. So now 46 chromosomes will be pulled to this pole of the cell, and then 46 chromosomes will be pulled to this pole of the cell. So in anaphase, you actually have 92 chromosomes temporarily. And then since you pull the chromatids apart to become individual chromosomes, you no longer have any chromatids in anaphase. The last phase of mitosis is called telophase. And then there's a lot of things going in telophase, but the one prominent feature of telophase is that you get the formation of what's called a cleavage furrow. So this is a cleavage furrow. This is a cleavage furrow. And then this cleavage furrow is just, why it's there is because it's just pinching off to separate this daughter cell from this daughter cell. And then we also say that telophase is the opposite of prophase because you get the reformation of the nucleolus. So this daughter cell will reform the nucleolus. This daughter cell will form or reform the nucleolus. And then you get the appearance of the nuclear envelope or nuclear membrane again here. And then also for this daughter cell. And then also your chromosomes, they will start to uncondense, uncoil to become chromatin again. So we'll have chromatin, chromatin. But since in telophase, these two are still connected to each other, where we're going to have 46 chromosomes here, 46 chromosomes here, we do temporarily again have 92 chromosomes and 0 chromatids in telophase. Now, the next process is called cytokinesis, and then cytokinesis does start at the same time as anaphase and telophase, but it is not a part of mitosis. It's a separate process. So cytokinesis is basically the division of the cytoplasm, which just means that this cell will just separate to become individual daughter cells through cytokinesis. So here we'll have 46 chromosomes for a total of 92 chromosomes in telophase. And then after cytokinesis, you'll have this daughter cell that will have 46 chromosomes. This daughter cell will have 46 chromosomes that will be genetically identical to each other. And then this picture, it just explains a little bit more about cleavage furrow. So the cleavage furrow, it is complete. It's completing in cytokinesis. So for cytokinesis in animal cells, we have the cleavage furrow, and then the cleavage furrow is just a ring of microfilaments. So remember, that's another type of cytoskeleton. And then what just happens with this ring is that it just gets smaller and smaller, just to pinch off and separate these two cells. So it'll separate this cell into two separate cells through that ring of microfilaments. For the plant cell, it's a little different for cytokinesis because the plant cell has a special structure, that cell wall. So what happens is in the plant cell, after it's gone through mitosis, it'll have these vesicles from the Golgi apparatus that contains cell wall material, which is just cellulose. And then these vesicles will fuse together, elongate, and then it'll eventually become the cell wall. And then the cell wall, so each daughter cell will have its own cell wall. So here's one daughter cell that will be genetically identical to this daughter cell. Cycle and their regulation. So cell cycle control system is just these sets of checkpoints that the cell has to go through to make sure everything is okay before going on to the next part. So we have a few of them. We have our G1 checkpoint, and then this is the most critical one. So during this checkpoint, this is the one that the cell will check to see if there's room to grow, if the cell size is correct, and then if the cell has enough nutrients to go ahead and prepare for mitosis and to even go through mitosis and cytokinesis, of course. And then we have to check for the presence of growth factors. So growth factors are chemicals. Some can be proteins, some can be estrogen lipids that will allow the cell to grow, to say, hey, it's okay to grow. If it's present, then the cell will grow. That's really important because you do not want cells to divide on its own accord. You want some kind of signaling factor, like a growth factor, to tell the cell it's okay to divide. You need to divide now. And then also during G1, the cell will check for any DNA damage, if there's any mutations or if there's damage to the DNA molecule, the DNA will hold, stop, and arrest, stay in G1 until it's able to repair that DNA, because you don't want to go into the S phase, DNA synthesis phase, and replicate that DNA that is not correct. You want to fix that, correct it, and then go on to S phase. So this G1 is basically a commitment state or a commitment checkpoint. Everything, if everything is okay, then the cell will go on to the rest of interphase and then, of course, the mitotic phase. But however, if the DNA can't be fixed, the cell will go into a G0 phase, which is the resting stage, which is the non-dividing stage, and just carry out its function for the rest of its life. So if G1 is great, if everything's okay, then the cell will proceed to go onto the S phase, duplicate that DNA, and then go into G2, where there's another checkpoint where the cell will check for its size, if it still has enough room to grow, and then check to see if the DNA replication is correct. Okay, so here, this is, an example of what can happen during G2. So here we have a healthy cell, okay, is undergone S phase, and then now is in G2 phase. So apparently there's some DNA duplication errors where it may have mistaken a nucleotide and put another nucleotide in. So the DNA, or the cell, it will attempt to repair the errors, and then if it's able to repair those DNA errors, great, then it'll go into the mitotic phase, mitosis and cytokinesis and complete cellular division. That's great. But if the cell is not able to correct those errors, those DNA errors, then what happens is the cell will undergo what's called apoptosis. And apoptosis, you can think of it as cell program, cell death. It's when the cell will kind of form into blebs and dispose of itself. Okay, and then so here, after it's formed these blebs, your lysosomes will come and take care and break all these blebs down. So here, this last checkpoint is called the M checkpoint, and it's at the end of metaphase. Is what we've talked about before as well. So here, the cell will check to see if the microtubules or the spindle fibers are attached to the kinetochores correctly, tightly. That's on the chromosomes. And then it'll also check to see if the chromosomes are aligned correctly on that metaphase plate. And then if it is, then it'll go through Anaphase, Telophase, and Cytokinesis. So there's other factors that affect cellular division. One, we talked about just now are growth factors. These are chemicals, substances that will kind of signal the cell that it's time to divide. And if there's no growth factors available or present, then the cell doesn't divide. It'll just go and carry out its normal functions. So another factor that will affect cellular division is something that's called anchorage dependence. So anchorage dependence is when the cell needs some kind of surface to divide on. In the lab, it's this plastic flask that the cell will adhere to, and then it'll start to multiply and divide. But in the cell, it can be the extracellular matrix that the cells will attach to and then divide. That way we can form tissues. Or the cell will actually, sometimes cells will divide on each other like a uniform single layer, and then you'll have multiple layers of cells. And then the last factor that we're going to talk about is called density-dependent inhibition, or also is called contact inhibition. So this is basically saying that when the cell has enough of a population, then it won't divide. It's just, crowded cells, they will not divide, because if it's crowded, if the density of the population is to its full capacity, then there's no room for the cell to divide. So in this picture here, we see that this flask here, is at full capacity. And then if you take out or remove a part of the cells, that population, then there is room to grow. These cells will divide until they're all touching each other. So this is this is how to regulate normal cells. But however, in cancer cells, when they start dividing, they don't need growth factors to be present to divide. They'll grow on its own accord. They'll divide on its own accord. Also, they don't really care about having, anchorage dependence. They don't really care about having a solid surface to adhere to. And also, they don't care about if the density is overpopulated or if they're, in contact with each other. They'll just divide, and divide. There's no regulation, basically. And then these cancer cells, they'll oversee those checkpoints. They will surpass those checkpoints that we talked about, that's supposed to regulate their cell division, but it doesn't. And then so they just keep growing and growing. So cancer cells, basically, they're cells that don't abide by the normal regulations that healthy cells abide by, like those checkpoints, those anchorage dependence, or that contact inhibition, is when their cell cycle doesn't function normally like a healthy cell. And then what you get is a growth of a tumor. And then tumors we've heard about are these abnormal masses of cells where they just keep dividing and keep dividing. But you have two categories of tumors. You have what's called a benign tumor. And then these benign tumors usually do not spread. They start off in one place and then they basically just stay there. But that doesn't mean that they don't have the potential to spread. So if they spread, that means that we say that the tumor has metastasized. The cancer cells has metastasized. That means they've broken away from their original site and moved into our bloodstream or our lymphatic stream, and moved to another part of our body. And then these tumors are usually malignant. So here we have an example. This picture's in your book. So here the tumor originated in the breast. And then apparently this tumor has broken or entered into the lymphatic system, which is a network system that has lymph fluid. And then the lymph fluid is where your white blood cells reside in. They travel all over your body and then to fight infection. And then of course it's a system where we can get rid of toxins and waste. So this is one way tumor cells can go and spread or metastasize, or they can go into our blood vessels, our bloodstream, break away. A part of the tumor can break away and metastasize. So breast cancer can metastasize into the liver organ. We've heard of those stories before. So how do we treat cancer these days? There are some new methods, but two of the oldest methods that we have come to know is radiation and chemotherapy. So radiation is just a beam of radiation, and these can include X-rays and gamma rays. And then what it does is that it destroys the DNA in those cancer cells so that they can no longer divide, so that they can no longer sustain life. And then you can have a beam of radiation for local tumors, or you can be put into a machine that will have radiation going from different directions. But we know that radiation can cause some side effects. So, some sterility, some nausea, vomiting. And then also the same with chemotherapy, where you can have some adverse side effects as well. We know that chemotherapy can cause you to lose hair, not make you feel well. You feel lethargic and just very low energy. So chemotherapy, you can have infusion of drugs to treat tumors that have spread. And then chemotherapy, certain drugs will target certain phases of the cell cycle. So, or certain components of the cell cycle. So you can have some that will target prophase, and then you can have some that will target the microtubules that are responsible for guiding these chromosomes. Okay, so now let's talk about some new terminology words that you guys will need to know for meiosis. We have what's called homologous chromosomes. Homologous chromosomes. Homo means the same, okay? And then it's just a pair of chromosomes that are very similar. So they can be similar in length. So here's a pair of homologous chromosomes at chromosome 1. You see that they're about the same length. Their centromere location is the same. They will have the same genes. And then also their staining pattern is the same. So here's a pair of homologous chromosomes, a pair of homologous chromosomes, a pair, etc. But your sex chromosomes, so if you're female, you're XX. That's, no doubt homologous chromosomes. They're exactly the same chromosomes. But if you're male and you're XY, well, these sex chromosomes may not be a matched pair or similar, but they function as a pair of homologous chromosomes. they function together. Some other words are locus. So on your chromosomes, on each chromosome, you will have certain genes. So here on this chromosome, the locus is for the gene for eye color. But you can have different versions or variant versions of that gene. So for example, we can have the allele for brown eye color. And then that can come from your mom. And then here from your dad, you have on the same locus the gene for eye color, but it may be for blue eyes. So we say that you have the father has the allele for blue eyes. And then here we have the locus for skin color. So it could be for dark skin color. But on the other homologous chromosome, we have the locus for light skin color. Or we say that you have the allele for light skin color. And then here, locus for hair color, it could be the allele for brown hair color. But here from the dad, we can have the allele for blonde hair color. So you see that this pair of homologous chromosomes, they have the same genes at the same loci. So loci is plural for locus. Loci is L-O-C-I. It has the same loci for the same genes, but we can have different alleles for those genes. And then we see that the homologous chromosomes can look like this, where the chromosome was duplicated. Okay, so remember that this is a sister chromatid, this is a sister chromatid, and then they are exact copies of each other. This is a different sister chromatid, and then this sister chromatid is an exact copy of this sister chromatid right here. So this is when the DNA has replicated, and then here, this is when the chromosomes have not duplicated. So here, this is a pair of homologous chromosomes. So this can be from mom, this can be from dad, from mom, from dad. So here in a normal karyotype in our body cells, our somatic cells, we have 23 pairs of homologous chromosomes. So if you count it, here we have 22, and then our sex chromosomes, that's 23 pairs. There's 2 in each pair. And then 22 of those pairs of homologous chromosomes are called autosomes. And then these autosomes are what you find in every male and female. And then of the 23 pairs, 1 pair are sex chromosomes. So here, I've talked about how females are XX. And then, but this picture, we see that it's a male because it's XY. So we have 23 pairs of homologous chromosomes, but 46 chromosomes total. Meiosis is for sexual reproduction so that we can make genetically different gametes. And then gametes are our sex cells, which are the egg cells and the sperm cells. And then this is what I normally would write on the board. So let's talk about the summary of meiosis. So meiosis, we start off with our diploid cells, which are 46 chromosomes, and then we undergo DNA replication or duplication, but only once. Even though meiosis has 2 cellular divisions, the DNA only replicates once. So that's important to remember. And then after doing so, what we end up is each chromosome will have 2 sister chromatids, but we still have 46 chromosomes, but we have 92 chromatids. And then we undergo meiosis 1 to get 2 haploid cells that will have 23 chromosomes, but they're in this duplicated state. So each cell will have 46 chromatids, 2 chromatids per chromosome. So 23 chromosomes, 46 chromatids, 23 chromosomes, 46 chromatids. This duplicated state is still too much DNA. So we have to undergo another cellular division called meiosis 2. And then these cellular divisions, they're denoted by the Roman numeral 1, 2. So after meiosis 2, each of these cells will produce 2 daughter cells that will be haploid still. So each will have 23 chromosomes, but the chromatids have split. So there are 0 chromatids, and it'll look like this. And then, so what happens to this daughter cell will have to happen to this daughter cell. So it will produce 2 daughter cells, or 2 haploids, that will have 23 chromosomes each, 0 chromatids each. And then overall, with meiosis, you produce 4 haploids that will have 23 chromosomes, 0 chromatids, that will be genetically different from each other due to an event called crossing over, which we will discuss later on. So these 4 haploid cells, or 4 gametes, can be 4 sperm cells or 4 egg cells. And then what happens afterwards is that the egg cell, which is haploid, that has 23 chromosomes, it'll unite with a sperm cell that is haploid with 23 chromosomes. They undergo fertilization to make a zygote to restore that 46 total chromosomal number, which is which is diploid. So in the beginning of this video, I told you guys that you will have to compare and contrast mitosis versus meiosis. So here, this is a chart that I've made to kind of outline some of the differences of both processes. So just, either copy it down or look at it, read it. But, it's something that, will, of course, mitosis we talked about. One thing that we haven't talked about in meiosis is that, so here, meiosis is to make sex cells. So we have an egg cell and a sperm cell. So here we have 23 individual chromosomes, 22 autosomes, 1 sex chromosome for this egg cell. The sperm cell has 23 chromosomes. It has 22 autosomes and 1 sex chromosomes. And then when you put these two together, you have your somatic cell, which will divide through mitosis. And then so here, the somatic cell has 23 pairs of chromosomes, 22 pairs of autosomes, and 1 pair of sex chromosomes. Okay, so now let's start on the stages or phases of meiosis. So meiosis is very special, it's very different. For meiosis, all the phases, you have to indicate the Roman numeral after these phases, because if on the exam, if you just put prophase, then we're going to think that you're talking about mitosis. But if you put prophase 1, we will know that you're talking about meiosis 1. If you put prophase 2, then we're going to know that you're talking about meiosis 2. Okay? So here in prophase 1, there's a lot of things going on. A lot of things that's going on here is basically the same as prophase of mitosis, where you have your nuclear envelope dissolving, that nucleolus is dissolving, you have the formation of your mitotic spindles, you have your centrosomes moving to the opposite poles of the cell, you have your chromatin condensing into chromosomes, okay? So all that is the same thing, but there's a special event that happens in prophase 1. And then this picture will kind of illustrate that. So here, what happens in prophase 1, the homologous chromosomes, they will find each other and start to pair up. So this can be from mom, this can be from dad, okay? So they come together and then they form what's called a synapsis. A synapsis. So to synapse means to touch to form a junction. And then what you get is what's called a tetrad. Tetrad. So tetrad means 4, and it's just 4 chromatids coming together. So here's one chromatid, here's one chromatid, here's another chromatid, and then here's another chromatid. So now what's going to happen is that these 2 non-sister chromatids are going to cross over and exchange DNA parts, DNA segments. So this chromatid from this chromosome will cross over with this chromatid from this chromosome, and then they will exchange nucleotides, DNA segments. And then essentially what you get are going to be 2 different homologous chromosomes. And then after meiosis 2, you'll have one haploid cell that will have this DNA, another haploid cell that will have this DNA, another haploid cell that will have this DNA, and then another haploid cell that will have this DNA. So these sister chromatids will separate, these sister chromatids will separate, and if you can imagine, that means that all of these haploid cells will have different DNA. That's why in meiosis, you form 4 genetically different haploid cells. And this is the main reason why you are different from your siblings. Another reason, you are different from your parents as well. Your siblings, they got the same DNA from your parents, or they got their DNA from the parents, but it's different because of this crossing over event. So in prophase 1, in humans, we have 46 chromosomes, but those chromosomes are in that duplicated state where each chromosome will have 2 sister chromatids. So that means you have a total of 92 sister chromatids in this prophase. And then in metaphase 1, you're not really doing anything with these chromosomal numbers or the sister chromatid numbers, so they stay the same. You're basically aligning these homologous chromosomes, these pairs of homologous chromosomes, on that equator in the middle of the cell, the center of the cell, or that metaphase plate. We can say that we are aligning the homologous pairs of chromosomes, or we can say that we are aligning tetrads. So this is a tetrad, this is a tetrad, this is a tetrad on that metaphase plate. And then it's really important that you compare metaphase 1, how it looks right now, to metaphase of mitosis. So here in metaphase of mitosis, we don't have homologous chromosomes. We just have chromosomes. And then next we have anaphase 1. And then what happens in anaphase 1 is that these pairs, these homologous chromosomes, they will separate. They will separate. So this homologous chromosome will go to this pole of the cell, and then its homologous chromosome, its mate, will go to this pole of the cell. So eventually what you're going to have are 23 chromosomes that will go to this pole of the cell, 23 chromosomes that will go to this pole of the cell. But you'll have 46 chromatids here, 46 chromatids here. Now this anaphase 1, since our sister chromatids don't separate, it means that these centromeres do not separate. But if you compare anaphase 1 to anaphase of mitosis, the centromeres do separate because the sister chromatids separated to become individual chromosomes during anaphase of mitosis. So in this phase, we'll have 46 chromosomes go to this pole, 46 chromosomes go to this pole. And how many chromatids do we have? 0. 0. But here in anaphase 1, we do have 92 sister chromatids. And then the last part of meiosis 1 is called telophase 1. And then in this one, you get the appearance or the prominent appearance of the cleavage furrow. So that's the same thing as telophase of mitosis, but the numbers are going to be different. So here you have 23 chromosomes, 46 chromatids will be in this part, and then the same thing, 23 chromosomes, 46 chromatids in this part. And then telophase 1 is going to be pretty much the same as prophase 1, where you're going to have that nuclear envelope will start to reappear, come back. Your nucleolus, even though you don't see it, it will start to reappear. Your chromosomes will start to uncondense back into chromatin. And then we're going to undergo cytokinesis, where this cleavage furrow is going to finish, and we'll have that contractile ring of microfilaments to separate these two cells. So eventually you're going to have 2 haploid cells that will have 23 chromosomes each, but 46 sister chromatids each. And then you notice that these chromosomes, they're still in that duplicated state where the sister chromatids have not separated from each other. So that means that that's too much DNA, and then we will need to undergo another cellular division called meiosis 2. So in meiosis 2, it's pretty much the same thing as mitosis, except the numbers are going to be different. But it'll look pretty much the same thing as mitosis. So here in prophase 2 of meiosis 2, we have our 2 daughter cells, and what happens to one daughter cell will have to happen to the other daughter cell. Each daughter cell will have 23 chromosomes, 46 sister chromatids. So in this prophase 2, we'll get the disintegration of your nuclear envelope. You'll have the disintegration of that nuclear membrane. You'll have the formation of the mitotic spindles. The centrosomes, they'll start to move to the opposite poles of the cell. And then in metaphase 2, you're not doing anything to the numbers. You're just basically aligning the chromosomes on the metaphase plate. But notice how metaphase 2 looks a little bit different from metaphase of mitosis, where we had that crossing over event. So each sister chromatids are not exactly the same anymore, like they would be in metaphase of mitosis. And then next you have anaphase 2, where the sister chromatids separate. So if they separate, that means the centromeres, the band, the region that was holding these sister chromatids together have separated, have split. So now these sister chromatids have become individual chromosomes, where you'll have 23 chromosomes will be pulled by those microtubules to this side of the cell, 23 chromosomes pulled to this side of the cell. And then you have telophase 2, which you'll have the prominent appearance of that cleavage furrow. And then you'll have the reformation of that nuclear envelope, reformation of the nucleolus. The chromosomes will uncondense or loosen back to chromatin state. Okay, so you have 23 chromosomes, 0 chromatids each. And then next you have cytokinesis, which is going to be separation of these two daughter cells. Cytokinesis, we usually don't denote a Roman numeral after that. So here we have two daughter cells that will have 23 chromosomes each. Here, if you imagine, we'll have two separate daughter cells with 23 chromosomes each. And then each of these daughter cells will have different DNA, will be genetically different from each other because of that crossing over event. So we can have four genetically different sperm cells or 4 genetically different egg cells. So this is a chart that I found online, and I think it's a very useful chart. So if you have time, please go over it. It just helps to compare and contrast the two processes. Okay, so there are 4 ways to increase genetic variation, genetic diversity. One of the ways that we talked about is crossing over that event in Prophase 1. So that's how you get different siblings. No siblings will be exactly alike. And then the ultimate source of genetic diversity are mutations. So mutations are spontaneous changes in the nucleotide DNA sequence. Another way to increase genetic diversity is that fertilization is random. So at any given point, a sperm with its unique DNA can fertilize any egg. So because the sperm fertilized the egg at a certain time, you were born. However, if it was at an earlier time or a later time, you may not have been born. Your sibling could have been born. And then another way to increase genetic diversity is something that's called independent assortment. And then this happens during Metaphase 1. So what happens in Metaphase 1 is you have these homologous chromosomes, these pairs, they line up on that metaphase plate. And then depending on how they line up, it will result in different haploid cells. So what do I mean by that? So you can have different possibilities of how these homologous chromosomes line up. So here on this side, we have maybe the blue chromosomes can come from dad, and then the red chromosomes come from mom. But keep in mind that this picture doesn't show the crossing over event. So here, if we have dad chromosomes, mom chromosomes, okay, in Metaphase 1, this is how they will line up. And then they're going to separate in Anaphase 1. And then this is how they will line up in Metaphase 2. And then in Anaphase 2, the sister chromatids will separate. And then you get these haploid cells. And then, of course, these sister chromatids will separate. separate, and you get these haploid cells. Now, if the homologous chromosomes were to align in a different way, such as here you have the chromosomes from dad right here, and then right here, and then the chromosomes from mom right here and right here. So this is a different alignment in metaphase 1 than this arrangement here. So these will separate from its homologous chromosomes in anaphase 1 to be lined up in metaphase 2 right here. These will separate from its homologous chromosomes in anaphase 1 to line up in metaphase 2 here. And the sister chromatids will separate. So this sister chromatid will go to this haploid. This chromatid will go to this haploid. And then the same thing happens to this cell, where this chromatid will go to this haploid, and then chromatid will go to this haploid. And you see how these haploid cells are different from these haploid cells. So these arrangements on that metaphase plate during metaphase 1 is random. It can be aligned in many different ways. So there are many different possibilities for different children with different traits. And then there are accidents that can happen during meiosis. One of the accidents is called non-disjunction. So non-disjunction is when the chromosomes or chromatids, they don't separate properly. And then depending on which phase it happens, the results can be severe. So let's take a look at this scenario. So here in anaphase 1, we know that homologous chromosomes are supposed to separate. Okay, so this pair separated properly. However, this pair did not separate, and we get non-disjunction happening to this pair of homologous chromosomes. So now it goes through meiosis 1, it finishes, and it enters meiosis 2, and then what's going to happen is that these chromosomes will go to this daughter cell, this chromosome will go to this daughter cell. And then in anaphase 2, all the chromosomes, all the chromatids, they have separated properly, but you have extra chromosomes in this daughter cell, whereas you're missing 2 chromosomes from this daughter cell. So if you're missing 2 chromosomes, we can only give this chromosome to this haploid cell, this chromosome to this haploid cell. So each of these haploid cells are missing 1 chromosome each. So remember, our N number is 23 for haploid cells. So 23 minus 1 is 22. So this gamete will only have 22 chromosomes. This gamete will only have 22 chromosomes. So where did the chromosomes go? Well, they went to this daughter cell. So here, after anaphase 2, these chromosomes will go to this gamete. These chromosomes will go to this gamete, where they'll have 1 extra chromosomes each. So here, this one will have 24 chromosomes, 24 chromosomes, 22, 22. So essentially, if non-disjunction occurs early in anaphase 1, you will get 4 abnormal gametes, which means that you will have 0 normal gametes. However, if non-disjunction occurs in anaphase 2, it's a little bit better. Okay, so let's go through this. So here, this is anaphase 1, where the pairs, the homologous chromosomes separate properly. So this goes to one daughter cell, this goes to the other daughter cell. And then here we can see that this daughter cell, the chromatids separate nicely in Anaphase 2. So these chromosomes will go to this gamete, these chromosomes will go to this gamete, where they will both be normal and have 23 chromosomes each. However, in this cell, we have non-disjunction occurring in Anaphase 2. So what happens is that these chromosomes will go to one gamete, and then these chromosomes will go to this gamete. So this gamete is missing a chromosome, so it'll have only 22 chromosomes, whereas this one will have an extra chromosome, so it'll have 24 chromosomes. So if non-disjunction occurs in Anaphase 2, you'll get 2 normal gametes and 2 abnormal gametes. So this is a chart that I made for you guys. It's in that assessment worksheets folder. It is important. Fill it out, okay? So basically, I filled in some of it for you guys. And then if you have any questions, just feel free to email me. Aneuploidy, what that means is that it's just when you have an abnormal number of chromosomes, and then there are some syndromes that happens. So when aneuploidy happens, usually the pregnancy ends up in a miscarriage. It'll abort itself. But depending on which chromosome it occurs on, sometimes the fetus will be, or the embryo will be viable. And then we get, the fetus, and then the baby is born. But the baby will have some unhealthy characteristics, will be what we say, not normal characteristics. So one of them I know everybody's heard of is called Down syndrome. And then there's another name for Down syndrome, which is called trisomy 21. So it just means that in a person that has Down syndrome, they have 3 chromosomes at the 21 chromosome. So normally you're supposed to only have 2 chromosomes. So you see, each of these chromosome positions, you have 2 chromosomes each. But here there's an extra chromosome. And it could have happened, the non-disjunction could have happened in Anaphase 1 or Anaphase 2. So if you have trisomy, which is 3 chromosomes, tri means 3, you have a total of 47 chromosomes. And then since it's on position 21, it's an autosomal chromosome. It happened on an autosomal chromosome. And then people with Down syndrome, their characteristics are, some mental retardation, some sterility, and then you have some facial features that will be different from ours. But that's okay because we have models that have Down syndrome, which is great. And then I do want to say that Down syndrome, the non-disjunction, it can occur from the father or from the mother. When people think of Down syndrome, they think it's always from the mother because after a female reaches a certain age, after 30 or 35, the chances of having a Down syndrome baby significantly increases, which is true, but, it can also happen occur because of the male sperm. So that's one syndrome. Another syndrome is called metafemale syndrome. And then normal females will have 2 X chromosomes, but in this syndrome, this female will have an extra X chromosome. So that means that they are trisomy. They have 47 total chromosomes. And then another name for it is called the triple X syndrome. So females that have this syndrome, the metafemale syndrome, they may appear normal, but some can have the characteristics of learning disabilities, muscle tone deficiency, and even motor skills development that are slowed down or that don't develop as quickly as a healthy person. Another syndrome is called Klinefelter syndrome. So a normal male would have XY. A person with Klinefelter, this male would have an extra X, an extra X. So that means that Klinefelter syndrome is trisomy because there's 3 chromosomes for a total of 47 chromosomes. And then since it affects the sex chromosomes, it's the sex chromosome. And then these males that have Klinefelter syndrome, they can have these characteristics where they are more female-like, where they'll have more fat, all around, even in their breast tissue. They'll have less facial hair, less body hair, small testicles. And then usually these Klinefelter syndrome patients, they will be sterile as well. And then we have Turner syndrome, which only affects females. And then this Turner syndrome, these females, they're actually missing an X. So a normal female will be XX, and then this O denotes that it's missing an X. So if Turner syndrome is missing an X, they will have a total of 45 chromosomes. 45 chromosomes. Their genetic makeup would be XO. And then it affects the sex chromosome. Sex chromosome. So these people with Turner syndrome, they'll basically look like this, where they'll have like a webbed neck. They may have heart problems, short stature. Some can have learning disabilities, mental retardation. And then the last syndrome that you guys need to know is called Jacob syndrome. And sometimes it's referred to as super male syndrome. It just basically means that they have an extra Y chromosome. So they are trisomy. It is affecting the sex chromosomes. And then they have 47 chromosomes total. And then Jacob syndrome is something that, is hard to tell if somebody has it. They can be taller. And then they may have some aggression issues. So based on those characteristics, it's hard to diagnose somebody with Jacob syndrome unless you do some kind of genetic testing. Okay, so that is all of chapter 8. I know that's a lot, but please take into account of everything I said and of everything I suggest for you guys to do well on this exam.